So today I'm going to be talking about riverine nutrient inputs and the extent of estuarine eutrophication in the Southern California Bight. This was a study done as a part of the Southern California Bight Regional Monitoring Program in 2008, and I would like to take this opportunity to thank or acknowledge my co-authors on this study. This was a large effort. We had 6 organizations that provided field team effort, and several individuals who helped prepare the study design and execute the analysis. So thanks to all of them: Martha Satula, Lillian Bousset, Sean Anderson, Shirley Brosick, Rosie Daggett, Jeff Crooks, Doug Gibson, Karina Johnson, and Lisa Stratton. Okay, so why do we care about eutrophication? We know that eutrophication is a leading cause of impairment in U.S. water bodies, according to the EPA. The main thought process for how this works is you have increased nutrient loading into the system, which results in a development of excessive organic matter production. And this leads to a cascade of effects, including dissolved oxygen, harmful algal blooms, and toxin production, as well as loss of habitat. And this translates into beneficial use impairment, which is where the monitoring comes in, where the rubber meets the road for management. However, we know that eutrophication is a problem, and there's a lot of data on a number of estuaries nationally. But if you look at the map of the United States, you'll notice that California has very little data on extent and magnitude. In fact, and in Southern California in particular, we only have enough data in 2 of the region's over 100 estuaries to make an assessment of extent and magnitude of eutrophication. However, nationwide, we do know that eutrophication is increasing, and that over half of the nation's estuaries are either moderate to severely impacted with respect to eutrophication, and this is actually getting worse in those systems. This is according to the 2007 NOAA National Estuarine Eutrophication Assessment. Okay, so now we know that there's a lack of data in California, and this study was designed to help fill that gap. So we had 3 main questions, and I'm going to center my talk around these 3 questions. The first being, what is the extent and magnitude of eutrophication in Southern California estuaries? The second being, what is the relationship between the expression of eutrophication and nutrient inputs? And finally, what are the implications for nutrient management in estuaries? Our study design was basically selected 27 segments from 23 estuaries in Southern California. They were randomly pulled from a master list of estuaries. We did provide some weighting for estuarine class so that we could actually capture a greater variety of estuarine types. And these classes were basically lagoonal systems that were either perennially open to tidal exchange or seasonally closed. We had river mouth estuaries that were both perennially open and seasonally closed, as well as open embayments. We monitored these sites from November 2008 through October 2009. It was a synoptic study of all these estuaries done about the same time. We targeted an index area within each estuary, and this index area was typically located near the freshwater source to the estuary, in an area of the estuary that we thought would have increased residence time. So if we were going to see symptoms of eutrophication develop in that system, it was going to be in that segment. So you can basically take the results from this study and assume that they're basically the most conservative answer for eutrophication that you could expect for these segments. Okay, so how did we decide what to monitor? Basically, there's been a movement towards using response indicators as the way to measure the extent of eutrophication in a system. And this is built around a conceptual model whereby you have limited nutrients in a minimally disturbed system coming into the system. In these minimally disturbed environments, you have a dominance of benthic diatoms or microphytobenthos and seagrass. You have light penetration all the way to the sediments, very good dissolved oxygen levels, nice sediment chemistry that's not toxic to benthic invertebrates. And then as you increase nutrient loading, you have a shift in the ecosystem. So this is a system that's now affected by eutrophication. The primary producer communities shift from seagrasses and benthic diatoms to these more opportunistic primary producers, macroalgae and phytoplankton. These are water column-based primary producers, so they tend to shade out the benthic diatoms and the seagrasses, so you have that loss of habitat there. The excessive organic matter production leads to respiration in the water column and reduced dissolved oxygen and pH as a result. And then you have changes in sediment chemistry due to this anoxic or hypoxic environment, which can include increased sulfide and ammonia, which is toxic to benthic creatures. So the types of indicators you can use to assess eutrophication in a system include the primary producer communities that are present, the water column chemistry, as well as the sediment chemistry. So those were the types of things we decided to measure in our eutrophication assessment. So our approach, in order to assess extent and magnitude of eutrophication, we did analyze a number of parameters, but the two main ones I'm going to talk about in this talk are the primary producer communities, which we assessed every other month, biomass and cover for the 1 water year for macroalgae and phytoplankton, and dissolved oxygen, which was measured using a data sonde in the bottom water continuously for the 1 water year. In order to establish a relationship with nutrient inputs, we measured ambient nutrient concentrations within the estuary. So these are water column samples taken from the surface of the segment right near the data sonde, and they were collected every other month, coincident with the primary producer measurements. And then the riverine nutrient loading was done via a combination of techniques, including continuous freshwater flow within the river, as well as dry weather concentrations, which were collected every other month, and MS4 data or model data wherever it was available. So, our first key question, what is the extent and magnitude of eutrophication in Southern California estuaries? And this brought us to the next major problem for our study was how do we make an assessment of eutrophication? You need to have thresholds, and the state of California is still in the process of developing criteria for nutrients for estuaries. So rather than develop our own, we decided to use a framework that was already available, and the one that we chose was the European Union Water Framework Directive. And the reason we chose this one was because it was based on indicators that we thought were most relevant to Southern California estuaries, namely dissolved oxygen, macroalgae, and phytoplankton. These were all elements in the framework. And they can categorize estuaries based on ecological condition, and they don't just give a black and white answer. They provide 5 different categories in which you can have, in which your estuary could fall into, estuarine condition, ranging from very high ecological condition, meaning near pristine condition, to very low, meaning it has severe impacts on the ecological condition. And in terms of the European Union Water Framework Directive, their mandate is that all of their water bodies in the European Union are going to have high or greater ecological condition within the next 10 years or so. So basically, that boundary between high and moderate is actionable in the water or the European Union. So we use this bright line, or we apply it to Southern California estuaries for better or for worse for this study. So I'm going to go through indicator by indicator and show you the results of the study. I'm going to start with the macroalgae assessment. So the macroalgae assessment is based on Scanlon et al., which is applied for or was developed for United Kingdom estuaries. And they basically categorize ecological condition using numeric thresholds for both biomass and percent cover, from very high ecological condition, low biomass, low cover, to very low ecological condition, very high percent cover and very high biomass. And down the bottom, I've showed you pictures of our study sites with their ratings. So you can see on the left here a segment that has very high ecological condition, low biomass and cover, all the way up to the estuary on the right, which has the highest biomass and cover of the sites we had measured in very low ecological condition. So just to give you a visual about what that means. So what were our results? We found that 78% of the segments we measured had macroalgae biomass and cover that was indicative of a degraded biological condition. I have here the map of the Southern California Bight, and each of the circles represents, some of the colors didn't translate, but basically we have colors here representing the value for each of those segments. And then I'm showing you a pie chart of the Southern California Bight estuaries overall, so you can see the relative percentage of estuaries in each of those categories. Now, we know from recent research efforts that it's not just the biomass and the cover that's a problem, it's also the duration of the bloom that affects ecological condition. In this case, this study was focused on benthic infauna. So here you have a figure from Laurie Green's PhD dissertation showing the abundance of sphenoids, which are a form of invertebrate found in a number of our estuaries, and they increase on this axis, and then treatments of algae that were applied to segments of the estuary ranging from 75, which, if you go back, was sort of a boundary here of a moderate condition, according to the Scanlon framework, up to 187 grams, which was a more poor, and 500 grams, which was sort of the maximum. And you can see that even for the moderate levels, the duration matters. After 8 weeks, you saw significant declines in the sphenoids at the lower end of the treatment, and for the higher treatments after 2 weeks. So the European Union framework does not incorporate duration, but we decided to investigate what that means for some of our estuaries, because we know that a number of our sites had excessive or extended periods of moderate or worse macroalgae biomass or cover. So this is a pie chart representing the number of estuaries in each of those segments. Only 11% had 0 periods of high biomass and cover, and then most of them had 1 or more. And here is how that looks in terms of biomass. Now, we measured every other month, so 4 weeks would be our first period, and then the second 2 periods of consecutive biomass would be basically 8 weeks. So this is how we're calculating that. Bloom duration may affect benthic and fauna in 59% of our segments, and an additional 30 may have biomass or cover, but because we didn't have higher resolution sampling, we couldn't discern it further. Okay, so let's move on to the phytoplankton assessment. The phytoplankton assessment is based on the annual average of continuous chlorophyll A data measured in micrograms per liter. Very high ecological condition, which would be a basically pristine type system, would be less than 5, and a very low would be greater than 30. And I have here what that data looks like. Now, you do expect, even in pristine systems, to have a period in the spring where you would have a bloom. Phytoplankton is actually very good for ecosystems. They're the bottom of the food web, so they provide food for all levels of the food web in the system. So most sites will have some sort of bloom in the spring. But the problem is when that bloom concentrations extend for a longer period of time, and then you start to have impacts on water clarity and effects on the ecosystem in general. So what did our assessment prove? It showed that about 39% of the segments had phytoplankton biomass that was indicative of a degraded condition, with most of the estuaries actually falling in this very high category. So phytoplankton was not a very problematic in a number of our sites. And this could be for several reasons, mostly or chiefly because they're very shallow systems that are subtidally driven. And I should point out that the major primary producer community is macroalgae in most of these systems, not phytoplankton. So 39% have phytoplankton biomass indicative of degraded condition. But again, it's not just the magnitude, it's also the duration. So there have been a number of studies that have looked at the effect of shading on the survival of seagrasses. And this was a study by Ruiz and Romero from 2001 that looked at the percent shoot survival here versus the number of days. And you can see that even after 2 months of continuous shading at 40% shade, that you have significant drops in the shoot survival, only increasing as the time increases. And at greater shading, it happens even sooner. So I'm going to use this 2 months here as a way to look at duration for Southern California Bight estuaries. So this is a pie chart looking at the bloom duration in each of our estuaries. So this is having that moderate or worse or moderate level of phytoplankton biomass for the annual average. So this is 24 hours of that of having excessive biomass, 2 weeks, 1 month, up to 2 months. So for 2 months, that's between 15% and then greater than that was 25%, where you would expect to have some effect on the benthic primary producers at that level. So dissolved oxygen. Dissolved oxygen was based on, or the dissolved oxygen assessment was based on the 10th percentile of hourly averaged dissolved oxygen data that was measured continuously from a data sonde. And the way you calculate this is basically you take your year's worth of dissolved oxygen concentration data and you rank it from the highest values to the lowest values and assign that as the percent of time. And then you draw a line at the 10th percentile, and that's the number that you apply the threshold to. And dissolved oxygen in this 10th percentile approach ranged from very high ecological condition or pristine, greater than 5.7 milligrams of dissolved oxygen per liter of solution, and then for very low would be less than 1.6. And recently there's been some effort for the state of California to define thresholds for California species, and it should be noted that the European values were very similar to those for California, so we think that this framework in particular is incredibly relevant for California estuaries. And using this framework, 75% of our segments had dissolved oxygen concentrations that were indicative of a moderate or worse ecological condition. And here says they fall on the map. And like the other two, it's not just the magnitude, it's also the duration of hypoxia. And if you look here, this is a figure from Vacquier, Sunier, and Duarte in PNAS in 2000 that looked at the median lethal time it took for a number of different species to have basically a failure point for that community. And the median was about 5 days, and then you have species at the low end and the high end. So there's a wide range in sensitivities to dissolved oxygen. But if you take that median range, you can start to look at what the longest single event would have implications for in terms of duration of hypoxia. So I'm defining hypoxia here as dissolved oxygen less than 4 milligrams per liter, and when a large number of systems here had a duration longer than 5 days, over a quarter. So the bottom line was that eutrophication is pervasive in Southern California estuaries. It didn't matter which indicator you used. There was still a significant number of estuaries that were falling in the sort of moderate or worse degraded condition category that would be actionable for the EU. All but one of the estuaries failed on at least one indicator, or not failed, but had a moderate or worse score. For macroalgae, again, 78%, phytoplankton, 39%, dissolved oxygen, 63%. And then there was a lot of discussion about how we might want to combine indicators into a multiple lines of evidence approach. And the assets framework, that's the assessment of estuarine eutrophic status, which was what they used in the NOAA National Estuarine Assessment. They used a primary indicator, which is a primary producer group, and a secondary indicator like dissolved oxygen to assign a value. And if you require that of our data set, then about 52% of our estuaries had moderate or worse condition for both the primary producer and dissolved oxygen. So basically over half of the estuaries are having a condition that might be considered degraded. Now, as with any assessment, there are a number of uncertainties that I wanted to make sure that were clearly defined here for this group. The first is whether or not our assessment was able to adequately capture variability. And there's variability on a number of scales. So essentially our study was a one-year synoptic study. We captured the seasonal variability fairly well by sampling every other month. However, we have no data on interannual variability. And you should note that the year we went out, 2008-2009, was a relatively dry year compared to other years in a 10-year period. So we're not entirely sure what that means for our assessment. The second scale is spatial variability. We took a segment approach to assessing these estuaries, but the scale is going to matter. Some of the estuaries were very small, and that segment basically encompassed the entire estuary. Other estuaries, like San Diego Bay, are very large, and the segment was only a very small portion of the overall estuarine area. So it's not clear how we would want to scale that to make an assessment framework for California. And then the second question is whether or not this was an appropriate assessment framework for the Southern California. Are these the right indicators? We think that answer is probably yes, they are the right indicators. Macroalgae was the dominant primary producer, and we did see response in dissolved oxygen. So, but are there other indicators that might be more relevant for Southern California or should be included in addition? And that I'm going to get to in the next session. And then finally, what is the relevance of the European thresholds? Are these the right, do they translate from a European model to a Southern California Mediterranean type model. And then there's very little guidance on how to integrate the data in order to get an answer, or that you would apply a threshold to. We decided to use the average period of highest biomass and cover, so that's the 2 consecutive periods of high biomass and cover averaged together to try to get a little bit of the duration of events incorporated into the model. But the Europeans just go out once, and they go out at the period that they think is the period of highest growth. But in Southern California, we had estuaries that had a bloom period in the winter, and others had a bloom period in the summer, and everything in between. So we need to determine for California what kind of guidance we want to have on how to establish timing and frequency of sampling. Now, we did do the assessment for how bad the system or the Southern California was using the European framework, but you can also use this data to just see how Southern California estuaries compare to one another. Now, to do this, we basically just took each of our primary indicators here and ranked them from highest biomass to lowest biomass, and then dissolved oxygen, lowest dissolved oxygen concentration to highest concentration, to categorize estuaries from highest condition to the lowest condition. What was the best estuary and what was the worst estuary, and everything in between. Now, to come up with the overall rank, we had to decide how we wanted to weight the different indicators. And we decided to go with the primary symptom, secondary symptom model, where we averaged the overall score, or the overall rank of the worst of the 2 primary producer indicators and dissolved oxygen, and that's how we came up with the overall rank. But more telling than even just the comparison of the different estuaries was how the indicators compared. And what I want you to see is there was often a discrepancy in what the indicators were telling you. So for Magoo Lagoon, for example, had very high macroalgal biomass and pretty moderate levels of phytoplankton biomass, but very good dissolved oxygen scores, which tells us something about how well the system was flushed. So yes, it has a high nutrient load, which is resulting in high primary producer biomass, but not necessarily degraded dissolved oxygen condition. And that was true for a couple of other estuaries as well. And then there were a few estuaries where the primary producers were sort of at odds. For example, perhaps the most dramatic was Santa Clara River Estuary, where it had very little macroalgae biomass at all during any season, but the worst phytoplankton biomass that we saw in any of the estuaries. So this was telling us that you could have primary producer communities that were basically at odds, where one was out competing the other, and they were giving completely different scores. So that gives us a little bit more information about where we want to go in terms of criteria as well. Okay, so that wraps up the extent and magnitude question. Basically, we know that eutrophication is pervasive throughout the Southern California Bight estuaries, although different estuaries are exhibiting different symptoms to different degrees. So the second question we wanted to explore was what was the relationship between the expression of eutrophication and nutrient inputs. So the first thing we wanted to do here was look at nutrient loads to see if we were actually capturing a disturbance gradient. And you can see here that these are the estuaries ranked from the furthest north to the farthest south, and the annual nitrogen load in kilograms. So yes, it looks like we did capture a fairly decent nutrient disturbance gradient in terms of estuarine loads. But we also know that estuarine size affects its ability to assimilate nutrient loads. For example, if you have an estuary like Mission Bay, which is relatively large, it should be able to handle a higher load of nutrients compared to Topanga Canyon, which is very small. So this led us to basically the whole idea of area normalized nutrient loads. And this is fairly common in the literature. But when you do that, you actually see that the loading picture changes, that there are some estuaries that are very small that are receiving maybe moderate loads of nutrients, but then when they're area normalized, they become incredibly high, and vice versa. So thinking about how nutrients assimilate nutrients is going to be very important in understanding or thinking about load-response relationships. Now, what do nutrient loads mean for estuarine concentrations? We found that when you look at nutrient concentrations in the estuarine water column, they were very highly correlated, or well correlated, with the nutrient load into the system, or significantly correlated, but fairly poor precision there. So loads are correlated with concentrations. So now, what does this mean for response? Basically, when we look at, this is annual macroalgae biomass, and the left panel here is the annual average nitrogen concentration in the segment, and then this panel here on the left is dry season macroalgae biomass versus the annual total nitrogen load that's been volume and residence time normalized. And you can see that nutrient inputs are significantly correlated with both nutrient concentrations and nutrient loads. However, the precision is quite poor. So there's obviously something else going on here. And for what it's worth, we think that this is actually mostly reflecting the disturbance gradient that we saw, because nutrient concentrations are definitely related to nutrient loads within the system. For dissolved oxygen, there was no relationship with nutrient inputs, either concentrations or riverine loads, the dissolved oxygen concentration is being driven by some other factor, which I'll talk about in a minute. But now it's worth asking, why is this correlation so poor? Why aren't we seeing a better response with nutrient loads or ambient concentrations? And this is basically because the ambient concentrations in both the estuary and the stream are what's left over. It's not a true representation of exposure. And if you look here, this is the estuaries from north to south again, and this is the percent of the total nitrogen concentration in estuarine concentrations. And you can see here, the yellow is dissolved inorganic nutrients, the pink is dissolved organic nutrients, or nitrogen, and the blue is particulate nitrogen. And you can see that the bulk of nutrients in almost every estuary, with a few exceptions, is comprised of either dissolved organic nitrogen or particulate nitrogen. And this is because particulate nitrogen is basically algal cells. You can think about that as directly being the macroalgae or the phytoplankton biomass. And the dissolved organic nitrogen is what's oozed out of macroalgae. Macroalgae, after they reach their peak, they tend to get a little bit leaky, and they leak out dissolved organic nitrogen, which is basically forming the bulk of the concentrations that we see in these systems. Secondly, the critical periods for loads are decoupled from the response. So for wet weather is dominating the loads of nitrogen into the system, and phosphorus too. So this is, again, the estuary segments on the left-hand column here, and percent of the annual tidal nitrogen load on the X, where the pink is dry weather, the blue is wet weather, and the yellow is just dry season dry weather. So critical period for nutrient loads is wet weather, but the critical period for each of our indicators is typically spring and summer. So here I have the percent of estuarine segments with peak biomass in each of the sampling periods, ranging from the November through September time frame. And you can see that most of them are peaking in the March, May, July, September time frame, so this spring through fall, non-wet season. Also, there's a whole nother source of nutrients in each of these estuaries that we haven't even accounted for when we're talking about riverine loads. And this is the sediment nutrients as a source to the system. And we can use sediment organic carbon as a proxy variable that might represent the contribution of sediment nutrients. And you can see here that the annual average of macroalgae and dissolved oxygen concentrations are significantly correlated with sediment organic matter. And that leads us to a new conceptual model for how we might want to think about nutrient loads and nutrient delivery into the system. So during the wet season, we'll have pretty large loads of both dissolved nitrogen and phosphorus, as well as particulate nitrogen and phosphorus. The dissolved nutrients will just get blown right out to sea with the high flows, but in particularly lagoonal systems, you'll have opportunities for deposition of the particulate NP. So that in the summertime, or the dry season, you will actually have some organic matter accumulation in the sediments that can act as a battery that can fuel algal biomass during the dry season, and that battery is going to get recharged every wet season. So when you get particulate deposition, organic matter gets remineralized in the sediment, and then the dissolved nitrogen and phosphorus can flux from the sediments into the water column, fueling this algal growth. So this is our new conceptual model for how nutrient delivery during the wet season might be fueling algal blooms during the dry season. But this battery, or the strength of the battery, is going to be different depending on the type of estuary. So the contribution of sediment nutrients is going to increase as the percent of fines increases, as well as the percent organic matter increase. So you're only going to capture this particulate nutrient form in a system that has much more of a lagoonal system setting, where you're not going to have the high flushing of nutrients and particles out of the system. So this on the left here is a study that was, or a combined set of studies here from myself and my supervisor, Martha Satula, from 2008, where we looked at a number of factors, including benthic flux, as well as a study by Will Barrelson, that we're looking at the percent of the total nitrogen load that's attributable to the flux of nitrogen from the sediments, and comparing that to the percent of organic carbon in the system. And you can see a fairly significant relationship there. So then you can use the sediment organic matter and fine-grained sediments as sort of a proxy for the contribution of benthic flux into the system. So over here I'm showing the percent organic carbon is highly correlated with the percent fine-grained sediments. And this brings us to the point that estuarine class is important. So there are some estuaries that are going to be more susceptible to eutrophication simply by the fact that their hydrology, physical habitat, and biogeochemistry is different. So for fluvial systems where you have very high water velocities, you're going to blow those fine-grained sediments right out to sea. So they're going to be dominated by sandy sediments. So you're going to have a much less percent fine-grained sediments. And then you're going to have low sediment organic carbon and low benthic flux. And then you're not going to have the battery of organic nutrients that can fuel algal biomass during the dry season. Whereas in more lagoonal systems, you'll have lower water velocities overall, you'll have much more fine-grained sediments, and then you'll have that battery recharged every year. So class is important when we're thinking about how we want to talk about eutrophication. So when we take all of our Bight 2008 data, and we try to fit a stepwise linear regression model to try to figure out which variables are most important to predict algal biomass or algal response. And for what it's worth, we did this for each of the three indicators, and we had similar results by and large. But for macroalgae, there were three main components that were important. And the most important one was this item we're calling diurnal Z, which is basically an indicator of the residence time within the system. So the longer the residence time, the more algal biomass we saw over and over again, and it was by far the most significant in terms of R-squared value, as well as p-values. The next most important predictor was organic carbon, with an R-squared slightly less, and then finally total nitrogen load alone was not significant unless it was area and residence time normalized. And when you put all three of these into the model, you get a pretty decent predictor for macroalgae response using the combination of hydraulic residence time, nutrient loads, and organic carbon. But still, maybe not quite precise enough to actually want to make or base regulations on. And I threw this in there just to give you an idea for how this actually looks in the field. So this is Loma Alta Slough in San Diego County. During its open state, it's pretty clean, or not pretty clean, but pretty low biomass. And then as soon as the inlet closes, you have macroalgae grow right up. So this is that whole residence time concept that might be helping to drive the susceptibility of eutrophication, in particularly in these systems that have a seasonal closure. So bottom line, nutrient response relationship is governed by site-specific factors. Across estuaries, nutrient loads and ambient nutrient concentrations are about equally correlated with response, and this is mostly reflective of the disturbance gradient rather than a true measure of exposure, since nutrient loads and ambient concentrations are correlated with each other. Across estuaries, estuarine class is driving a large component of the variability within the nutrient response relationship, primarily that hydraulic residence time. So I want to end here with some implications for nutrient management. So for managing eutrophication, we're assessing indicators of eutrophication, but you need to get back to nutrients somehow. And we made a really good effort of it during the Bight, or using the Bight 08 data set, by creating models that are trying to figure out what are the most important drivers between nutrient inputs and biological response. But there are a number of challenges here. So how do we want to make the assessment? Do we have the right approach from the Bight 08 study? How should it be modified? What indicators do we want to use? How do we want to integrate the data? Because we found that all of these things matter. For example, if we're in Santa Clara River Estuary, the one that had a very high phytoplankton response but no response in macroalgae, if we just use macroalgae, we're not going to capture the state of eutrophication in there. So you want to be sure that you're capturing the right indicators for the system. And then you also want to be able to incorporate the frequency and duration of the data properly so that you're adequately characterizing the system. And this is going to require some work on reference condition as well, because you would expect for an estuary that seasonally closes, even in a pristine environment, may have high algal biomass and low dissolved oxygen during part of the year. So we need to actually think about how we want to make the assessment and how we want to set those thresholds for these different types of systems. Then we should think about what you want the target to look like. Do you want it to be ambient concentrations, like the basin plan objectives? Should they be nutrient loads, like in TMDLs? But in this study, we showed that sediment nutrients are incredibly important in a number of systems as an additional source of nutrients to the system. So that would be something more like regulating particulate loads rather than just dissolved loads, or in combination with dissolved loads. And then finally, how do you want to incorporate site-specific factors like the depth, volume, and the residence time to make these associations more clear? So basically, in this study, we used statistical models to try to establish some relationships between nutrient input and site-specific factors and ecosystem response. But you could go all the way up to calibrated numerical models, like the ones that are often used for TMDL development, but these are incredibly expensive, and there are over 100 estuaries in Southern California alone, and if you need to put in a couple $1 million per numerical model, at some point you're going to get ridiculously cost prohibitive. So we need to come up with a better solution. These statistical models were very helpful at helping us understand what factors are important in terms of eutrophication, but they're not really precise enough to set a numeric target. So our next step here at SWRP is to try to think about development of simple box models that account for residence time and that build in some empirical relationships between organic carbon and benthic flux to try to help us assign a better value for a nutrient target rather than having to go the multi-million-dollar route for calibrated models for every system. So I'm going to end here with conclusions. Basically, eutrophication we found to be pervasive in Southern California Bight estuaries. Across these estuaries, we found that nutrient load and ambient nutrient concentrations were equally correlated with response, but that the biggest component of the relationship between nutrient loads and response was driven by estuarine class, specifically variables associated with residence time. And that these nutrient response models, if you want them moving forward, help setting numeric targets for things like TMDLs, they're going to need to incorporate more site-specific factors like residence time and benthic nutrient fluxes. So now I will be happy to take some questions. I'd like to point you to the Bight 08 Estuarine Eutrophication Assessment report, which is on SWRP's website. I'm also happy to receive any questions after today via email, and that's my email right there on the slide.